Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Okay, so for years, maybe decades even, when you thought about alcohol and brain health, there was this persistent idea, wasn't there?
Oh, absolutely. The idea of an optimal dose.
Exactly. Like maybe that one glass of wine or beer at night wasn't just, okay, it might actually be good for your brain. That famous U-shaped curve everyone talked about.
Yeah, the J-shaped or U-shaped curve. Observational studies seem to show it again and again.
Well, today we're digging into something that pretty much rips that curve right up. It's a huge new study, just out in BMJ Evidence-Based Medicine.
And it's really groundbreaking. It's the biggest study yet combining traditional observation with sophisticated genetic analysis to look at alcohol and dementia risk.
And the headline finding we need to unpack for you is, well, it's pretty stark. That protective effect from low consumption, it looks like it's probably just not real.
Yeah, the study suggests any amount of alcohol might actually be detrimental. The risk seems to just go up steadily the more you drink. No safe harbor, so to speak.
A really sobering conclusion, pardon the pun. Let's get into why the old idea was so common first.
Right. Well, the big issue with the older studies, the observational ones, was how they handled non-drinkers. It's a bit tricky.
Yeah.
They often failed to properly separate, people who'd never really drunk alcohol from those who used to drink, sometimes heavily, but had stopped.
The sick quitters idea.
Precisely. People might stop drinking because they're already feeling unwell, maybe even early. Very subtle signs of cognitive decline kicking in.
So if you lump those folks who might already be on a path towards health problems in with lifelong teetotalers.
Exactly. It makes the whole non-drinker group look riskier than it actually is.
Which then makes the light or moderate drinkers look comparatively better, right? Protected, almost.
That's the illusion. It's what we call reverse causation. The developing disease, or its early symptoms, is causing the change in drinking behavior, not the other way around. It messes up the statistics.
Okay, that makes a lot of sense. So this new study needed a way around that statistical ghost.
And they found one, using just incredible scale and a clever dual approach.
Talk about the scale first. Where did they get the data?
They used two absolutely massive health databases, the U.S. Million Veteran Program MVP and the UK Biobank, or UKB. Huge resources.
And how many people are we talking for the observational part?
Over 500,000, specifically 559,559 participants aged between 56 and 72 when the study started tracking them. Following that many people is impressive.
But that's still based on questionnaires, right? What people said they drank.
It is, and that's where the second part, the really innovative bit, comes in. They didn't just rely on self-report.
This is the genetic part.
Yes. They incorporated genetic analysis using summary data from, an astonishing 2.4 million participants. And they use a technique called Mendelian randomization, or MMR.
We hear MMR mentioned a lot now. Can you break down why it's seen as such a powerful tool, sort of simply?
Sure. Basically, it's the next best thing to a perfect long-term randomized controlled trial, which for alcohol is obviously impossible. You can't ethically tell people how much to drink for 50 years.
Okay.
So MR uses genetics as a stand-in, a proxy. We all inherit gene variants randomly at birth. Some of these variants are known to subtly influence how much alcohol a person is likely to consume over their lifetime.
Okay.
And because those genes are assigned randomly, they aren't linked to lifestyle factors that usually muddy the waters in observational studies, things like diet, smoking, income, education, all that noise.
So the genes provide a cleaner signal, less bias.
That's the idea. By looking at health outcomes, like dementia, in people genetically predisposed to drink more or less, MR tries to estimate the causal effect of alcohol itself, stripping away those confounders that created the misleading U-curve.
Got it. Okay, so this is where things get really interesting, the clash between the two methods. What happened when they just looked at the questionnaire data first? The old way.
Well, just looking at the phenotypes, the self-reported drinking, they found exactly what previous studies found.
The U-shape.
Yep, the classic U-shape. Compared to light drinkers defined as fewer than 7 drinks a week, the non-drinkers and the heavy drinkers, those having 40 or more drinks weekly, they showed about a 41% higher risk of dementia.
And what about people with diagnosed alcohol use disorder?
Their risk was even higher, about 51% greater than the light drinkers. So the old method confirmed the old story.
But then came the genetic analysis, the MR results.
And this is where the story completely flips. The Mendelian randomization analysis found zero, absolutely no evidence of a U-shaped curve.
No protective effect at low level?
None whatsoever. Instead, what they found was a consistent monotonic increase in risk.
Monotonic meaning?
Just a steady, straight line upwards. Basically, the higher your genetically predicted alcohol consumption, the higher your dementia risk. No diff, no protective zone, just more risk with more alcohol starting from very low levels.
Wow. Did they quantify that increase? Like how much extra risk per drink?
They did. The genetic model suggested that even a small increase, just 1 to 3 additional standard drinks per week, was linked to about a 15% higher risk of developing all-cause dementia.
1 to 3 drinks a week. That's not a huge amount. A 15% jump in risk associated with that is significant.
It really is. It suggests the harm is cumulative and starts early, without that supposed protective sweet spot.
So this brings us crashing back to the aha moment. How did the observational data get it so wrong for so long? It has to be that reverse causation thing again.
And this study provided really strong evidence for exactly that. They used the long-term health records from the Million Veteran Program, tracking people's drinking habits over many years before any dementia diagnosis.
And what did that tracking show?
It showed the sick quitter phenomenon in action, clear as day. People who eventually developed dementia tended to reduce their alcohol intake, sometimes quite significantly, in the years leading up to their diagnosis.
So the very early stages of the disease, the prodrome, were making them drink less.
That's exactly what it looks like. Maybe cognitive changes made alcohol less appealing or harder to tolerate, or maybe lifestyle changes associated with early decline played a role.
But the point is, they were already developing dementia, and they were drinking less.
Correct. So they end up in the low consumption or non-drinker group, making that group appear to have a higher dementia risk, which in turn makes the moderate drinkers look falsely protected. The illness was causing the reduced drinking, not the other way around.
That longitudinal data really nails it then. It shows the causal arrow is pointing the wrong way in those older studies.
It strongly suggests that, yes.
And this study looked at all-cause dementia, right? Not just Alzheimer's specifically.
That's right. Which is important because it gives a broader picture of alcohol's potential neurotoxicity. It's not just about one specific disease pathway. It seems to be detrimental across the board for cognitive health.
It really challenges that whole idea that a little bit might be good for the brain.
Fundamentally challenges it. The authors state clearly these findings undermine any notion that low alcohol levels are neuroprotective.
And the implications for public health seem pretty huge.
Definitely. If alcohol consumption, particularly problematic use like alcohol use disorder, increases dementia risk, then tackling AUD becomes a really important dementia prevention strategy.
Did they estimate the potential impact?
They did some modeling. It suggested that if we could somehow cut the prevalence of alcohol use disorder in the population in half, we might prevent up to 16% of all dementia cases.
15%. That's a massive number when you think about the scale of dementia globally.
It's a very significant potential public health gain grounded in this strong genetic evidence.
Now, we should always touch on limitations. Even massive studies have them. What did the researchers point out?
Well, one key thing is that the genetic analysis, the MR part, had the strongest statistical power in people of European ancestry. That's just because the big biobanks used, like UKB, are predominantly composed of that group.
So, generalizing the exact dose-response curve to all ethnic groups needs a bit more caution.
Yes, although they did find similar risks associated with diagnosed alcohol use disorder across different ancestry groups they could analyze. But the fine-grained analysis of consumption levels was most robust for European ancestry populations.
Anything else? MR itself relies on certain assumptions, doesn't it?
It does. Mendelian randomization methods have underlying assumptions about how the genes relate only to alcohol consumption and not other dementia risk factors directly, for instance. These assumptions are plausible, but can't be proven with absolute certainty.
Okay. But overall, the consistency across different genetic markers they used was pretty strong, you mentioned.
Extremely strong. They looked at genes related to alcohol quantity, problematic use, and dependence, 3 different angles. And the signal pointing towards increased risk with increased consumption was consistent across all of them. That really boosts confidence in the findings.
So let's boil it down. The main takeaway for you listening is, when scientists use these advanced genetic tools to cut through the fog of lifestyle factors, that old U-shaped curve disappears.
It seems to evaporate, yeah. What's left is a much simpler and perhaps more concerning picture, a steady linear increase in dementia risk with alcohol consumption. No apparent safe level, no protective benefit.
The illusion of the curve is replaced by the reality of the line,
A good way to put it. And it leaves us with a pretty critical question, doesn't it?
which is,
Well, if past research, even if flawed, led people and maybe even health advice to think light drinking was harmless or even beneficial, what now? Now that this very strong genetic evidence points towards a steady increase in risk from the start, how quickly should public health guidance adapt? What's the responsibility there? It's something important for you to think about.
a really powerful final thought. How fast does science translate into everyday advice when the message changes so dramatically? Thank you for joining us for this deep dive into the source material today.
Thanks for tuning in.
We'll see you next time.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.